Okay, so we are starting Unit 4 Outcome 2 today. And Unit 4 Outcome 2 mostly focuses on things related to genetic technology. So that covers a number of different things. We're going to start off by looking at the basic tools that scientists use to manipulate DNA, move into look a bit about how that's applied to things like GMOs. And then from there move into things like epidemics and pandemics, which is very relevant, antibiotic resistance, and a bit about further changes in biotechnology. But for today, we're just focusing on the basic overview of what the genetic tools are, and we'll look at two of them in detail, which are restriction enzymes and DNA ligase. So, as I said, we're going to look at various things that scientists use to manipulate DNA throughout this topic, and there's lots of reasons why we want to be able to manipulate DNA and do different things with DNA. So that can include pharmaceutical reasons, such as vaccine production. So in order to make vaccines, we need to be able to potentially isolate certain bits of DNA or mRNA. That can include agricultural reasons, so genetically modified organisms and transgenic organisms, which are things we'll look at later on. Those are both examples of ways that DNA have been manipulated. There are forensics, so that includes crime scenes, identifying of dead bodies and stuff like that. That all relies on the ability to get lots of DNA from a small sample and analyze that. It includes medical reasons, such as gene therapy and genetic testing, and also legal things as well, such as paternity disputes. So all of those would, be reasons we need to be able to do this. So there are lots of different tools and processes that scientists use to manipulate DNA. That includes having to be able to cut out specific bits of DNA, for which they use restriction enzymes, being able to join bits of DNA together, for which they need DNA ligase, being able to separate DNA fragments out according to size, for which they need a process called gel electrophoresis, the ability to isolate and identify specific bits of DNA, for which you need DNA probes, the ability to amplify DNA, meaning to make many more copies of DNA, for which you need the polymerase chain reaction, PCR, and then the ability to transport DNA through vectors. So today we're just focusing on the restriction enzymes we use to cut DNA, and the DNA ligase that's used to stick things together. We'll look at the others through further lessons as we go. So with restriction enzymes, they are sometimes also known as restriction endonucleases. That's just one of those things. Be aware that both may be used. Sometimes you'll hear restriction enzymes, sometimes you'll hear restriction endonucleases. So what these are enzymes that will recognize a specific DNA sequence and then cut at that site. And the way they cut can be either to cut making a blunt end when there is a straight cut down the at the recognition site, or with a sticky end, which is when there is either an overhang or an underhang. So I'll show you what I mean by that. So there, I've got a DNA sequence before being cut, and the recognition site is about here. So the blunt ends would be if the enzyme cut it just straight like that, so straight down there. Whereas a sticky end results in an overhang or underhang like here. So it cuts it in a way that a few bases are exposed here and a few bases are exposed there. And those are called sticky ends because if we try and join it with a complementary sequence, so I've got AA here, if we try to join it with a sequence that has a TT exposed there, theoretically it should stick to it, okay? It should bind with it, and that's why they're called sticky ends. So normally, these sticky ends are more useful than the blunt ends because the sticky ends can join up to other sequences of DNA, which, as we'll see throughout the topic, is fairly useful. So that just covers restriction enzymes. And like I say, restriction enzymes, they're there to cut out specific bits of DNA. They recognize it and they cut it, usually with sticky ends, so that we can use it to bind to other things. And then we also need to look at DNA ligase. So ligase is an enzyme that joins DNA fragments together. So it's almost like the opposite. Restriction enzymes, cut DNA. DNA ligase binds bits of DNA together. So yeah, the reason why it's able to do that is because DNA is a universal molecule. What that means is, while there are differences in sequences of DNA between different organisms, the actual structure of DNA is practically identical. It's always just got the sugar phosphate backbone and those 4 bases making up the middle, the A, T, C, and G. So DNA's got a universal structure. And what that means is we are actually able to insert a genetic sequence from one organism into another using both restriction enzymes and DNA ligase. That is important for gene cloning and bacterial transformation, which is something we'll look at in a later lesson. But I just wanted to give a basic overview of that now. So here I've got a bacterial plasmid. So a plasmid is a circular loop of DNA found in a bacterium, and I've got a gene of interest that's been isolated. And these have both been cut using the same restriction enzyme. So the plasmid has been cut open with the same restriction enzyme that cut this gene out of its DNA sequence. What that means, and if you look really closely, you can see I've deliberately drawn them with sticky ends. So this overshoots a bit, this undershoots a bit, and likewise on this side as well. So they've got sticky ends. Cutting something with the same restriction enzymes ensures the sticky ends are complementary. So basically, the bit that goes over here will be complementary to the bit that goes under here, and the bit that goes under here will be complementary to the bit that goes over here. Okay, so when we expose these to DNA ligase will basically stick them together to form a new plasmid that's got the gene that we were interested in inside of it. So that is something called bacterial transformation, which will be another lesson later on. But for now, just to be really clear, the main things I wanted you to get from this is understanding how the restriction enzymes or restriction endonucleases work. So remember, those are the enzymes that will cut DNA at a specific recognition site, either with blunt or sticky ends, and to get the idea of DNA ligase. So DNA ligase will bind separate strands of DNA together. So please do the work that's on Google Classroom as well, and as ever, if you need any help, log into Zoom throughout the session, and also make sure you are in Zoom at the time that I specify today as well. Thank you.